Castro, who's the organizer of our Desirable Tomorrows conference. And I mean, without much more, let's get started. So this is the fourth session, and today our focus is more on the living world. Desirable Tomorrows, in two short words, sums up very well what universities are about. We're about doing research, creating new knowledge, sharing that knowledge, with the desire for a better tomorrow. And the framework that we're working within today is within sustainability. And this panel, panel 4, is specifically focused on our pro-international activities. So UFRJ has an international, has an institutional international research program that is strongly supported by the federal government CAPI's print program. So we'd like to thank CAPI's print for their support. And over the last 3 years and for the next 3 years, we hope to mobilize 1,000 researchers, be they teachers, be they technicians on short technical visits, be they students on 6-month or 1-year placements. And the idea is to research and work together and answer fundamental questions that hopefully will give us a better tomorrow. Now, today I have 6 speakers for us, and they'll give relatively short talks. The first speaker will be Fabio Scarano, and I met him, and he may not remember this, in 1995 in the Royal Botanical Gardens in Kew. And that time I watched a talk on plant physiology. And then it's funny how academic worlds work. I was a PhD student at Newcastle University in England. I met a Brazilian, and I moved to Brazil and have been working and researching ever since in environmental microbiology, also in the biodiversity area. And I didn't really see very much of Fabio, and we all get busy. But I discovered later that he'd been very important and busy working with the United Nations, and it's a great honor to have him here today. I then saw him again about 3 or 4 years ago in an inaugural lecture for our Center for Health Sciences. And what I took from that message, which was the welcoming message to our brand new undergraduate students, was a message of love. That was the key word in that presentation. And I think that's the other, is what we want. We want less fear, more love, putting it very, very simply. I'd like to just read out Fabio's mini bio, just so as not to do him justice. So he is his graduation is in forestry engineering. He has a PhD in ecology and is a full professor at UFRJ. He's been actively involved in the United Nations groups to do with climate and biodiversity. He's been the director of Rio de Janeiro's Botanical Gardens, which is different, but I'd say almost the same or as good as Kew Gardens. He has written a number of books and has received 2 premios of the Jabuti for literature. His most recent book is called Regenerante de Gaia from 2019. So it's a great, that's Fabio. Before I rush ahead and get ahead of myself, we've got 5 other talkers too. So after Fabio gives us his talk, which will give us a vision of some of the opportunities and challenges that Brazil faces, because Brazil is a superpower in terms of biodiversity and bioculture. So Fabio is going to give us the big picture, and then we're going to move on. And we're also extremely lucky today to have a full professor, Adriana Silva Hemeli, who's going to talk to us. Now, you might not know, but she's also the coordinator for all biotechnology research, the federal government. But that's the sort of title you get for being an excellent scientist. And she's going to share with us a shorter talk about biotechnologies for sustainability. So as I said, our Desirable Tomorrow is about sustainability. We are interested in protecting the world and also making the world a fairer and safer and healthier place for everyone else. So she's going to talk about how we can feed the world better by improving plant productivity, which is food security, which is is within the first 3 objectives of the United Nations. We have zero poverty, zero hunger, and then quality of life. So we don't want, and the research that Adriana works with focuses on that. But not only are we going to learn from her a little bit about improving food security, we'll also learn how plant biotechnology and molecular biology and 5th generation industrial revolution generation new ideas can protect plants against or help plants lead with climate change. So almost like a fast forward evolution. So that'll be Adriana, and then after that, because we've got a great afternoon, we will have work presented from 4 of our ongoing PhD students that have worked in international partnerships, that have left Brazil and taken their research projects all over the world to try and help protect and make the world more sustainable. We'll have Vivian, who will be looking at a combination and the importance of water resources and forestry. We will have Bruno, and Bruno is going to focus on the microbial world and holobionts. He's going to look at sponges and tell us a little bit about the bacteria and what they do there. We'll have Marina, who's going to talk to us about conservation, and she's going to also bring about some ideas that are important, where the public has maybe failed to conserve, perhaps the private can. Private reservations. Let's see. I haven't heard her talk yet. And then we have Michele, and she's going to take us back to right to the beginning. Maybe we'll put Michele before, which is the importance of light, the energy that drives everything else that happens on our planet. And she's going to look at what happens with photosynthesis on microalgae. So we've got a full afternoon. I do hope that you will enjoy it. Feel free to send questions, write in questions, because we will have an opportunity to answer some of those. We'll also be, each of our speakers will speak in their native tongue. I'm originally from England, that's why I'm speaking in English, but I think most of our speakers will speak in Portuguese. Please make the most of the translate button that you've got on your screen. But so without talking anymore, let's ask Fabio. Fabio, please switch on your video. I can't see, but hopefully switch on your microphone. And let me say thank you very much for being here, for being able to give us this talk, and I'm sure we're all in for a treat. So I'll pass the palabra, pass the word over to you. Thank you.
Andrew, muito obrigado.
Thank you very much, Andrew. I'd like to thank you and the organizers for this opportunity to bring this presentation to this event, which is so important to our university, and sharing what we've been doing with colleagues from all over the world. I'm so happy to see you again. Sometimes I have the feeling that we met more often in Britain than in Brazil, but I think this has to do with the fact that when you came to Brazil, it was the time when the UFRJ was receiving me and let me go to some adventures outside the academia, and this is why we haven't met that often. I worked for the government, for NGOs, and what I will share with you today have to do with these experiences and the time I spent in the political area, for example, is something I can bring back to our students in the form of classes and research projects. I'm very pleased to see Professor Adriana. It's been a long time. We haven't seen each other for a long time. We had different moments at the UFRJ and at the Botanical Gardens in Rio. And my dear students, Viviane, Marina, who spent the beginning of the pandemic such a tough time around the world, and they were abroad because of this project that we have concerning internationalization, and they're back in Brazil. They're back in Brazil, and I'm pleased to meet Michelle. When you met me, I was working on photosynthesis, and my research has changed dramatically. Bruno sent us recording, but I hope he will be able to be present here. I'm bringing a more general message regarding Brazil. We are a biocultural superpower, and I will mention the opportunities and challenges that this brings to us in Brazil, as Andrew said before. I'd like to start by defining a biocultural superpower. What is this? We are in the more biological areas, and we speak a lot about biodiversity, the biological diversity. It's a curious thing regarding modern human beings. And when we talk about biodiversity, we exclude ourselves. It's like speaking about nature as if it were something different from ourselves. And deep down, biodiversity is one single thing. It has to be with nature, and nature has to do with culture as well. So I've been using this phrase, biocultural diversity, that has to do with all manifestations of diversity, biological diversity, cultural, linguistic diversity. And Brazil is a very interesting country in this context because it is one of the 17 megadiverse countries in terms of biodiversity. It's part of this, and the group of megadiverse countries are 17 countries that have 70% of the biological diversity in the world. And Brazil also has 300 languages spoken here. In the Brazilian Amazon, they speak 274. And at the UN, we speak 200 languages, and the Amazon has more languages than the UN. So this cultural and biological diversity, make Brazil a superpower. So the cultural diversity in the country reflects somehow its biological diversity and vice versa. This brings huge responsibilities to the country, both responsibilities and risks. And I would like to give you a global overview. And unfortunately, the more we advance in terms of biodiversity studies, the more we lose. We lose about 100 to 1,000 times more species than expected. We have 1 million species that are endangered today, according to the UN platform. And this leads a set of scientists to believe that we are undergoing the 6th wave of species extinction today. And of these 6, we are facing the single extinction caused by a single species, which is human species. And the diversity picture in terms of biodiversity loss is expressed in this map. The more the red you see, the more biodiversity is lost. Here in Brazil, especially in the center, as what we call Cerrado, our tropical savanna, and also on the coast, the Mata Atlântica system, have systems that have lost 30% of their biological diversity. The same happens in the Pampas and in Mexico, the US, and in various parts of Asia, China, Australia, Sub-Saharan Africa. We have many parts of the world that have lost their biological diversity. And what scares us is that in the light of the climate changes that are happening, what we project between 2080 and 2100, we project a very serious loss Today, we have 25% of the species that will be lost. So 1/4 of the species living in this continent are threatened and are endangered. Here in the northern hemisphere, the risk is smaller, and on the sea, we have about 15%. The loss of species, if we put aside all ethical concerns, if we think about, and if we have a utilitarian focus, this means a loss of means of life and wealth. Because of that, biodiversity is central to a set of global agreements that are enforced today, and many of them started here in Rio in 1992, and in an event the UN called the Conference on Humanity and the Environment, which became known as ECO 92, and two things were highlighted there: the struggling against desertification and global warming. We had targets for 2021-2030. We didn't hold that meeting because of the pandemic, and it will be held in China. And we will establish some goals to be reached until 2030. And we have targets to neutralize the degradation of the earth between 2015 and 2030. And Paris, many countries around the world signed the climate covenant. And until 2030, we will change the way we live. So in order not to overcome 1.5 degrees Celsius in temperature. And we would like to highlight that science is showing we are 1.1 degrees Celsius above, and 2 degrees above what we have will mean the polar cap melting and lots of problems in the coastal areas. So the science had a panel on the climate, and they believe it's been a good thing. 1.5 degrees is good, but 2 degrees is something we don't want to reach. So fighting degradation depends on biodiversity. The agreement of the countries in the climate convention counts on many solutions based on nature, and we will listen to Viviane talk about restoration.
Now, Michele will be talking about photosynthesis. So, we depend on biodiversity for this agreement to work. The Sendai mark has targets for 2030. So biodiversity is important to reduce disasters such as droughts, floods, landslides, and finally, the Sustainable Development Goals. This is a set of 17 objectives to be reached until 2030, and they were signed in 2015 in New York at the UN, but the discussion on them started also here in Rio de Janeiro in 2012 at the Rio Plus 20 event. The 3 meetings that originated here came together and wrote a letter called The Future We Want, which was a base for this instrument, this aspirational instrument, which are the SDG, the 2030 agenda. Notice that all the goals are here to be reached till 2030. In summary, we have 10 years, about 10 years, to change the world very much. These are the 17 SDGs, Sustainable Development Goals, which I'm sure you know very well. They combine social, economic, environmental, And these objectives are very well summarized in this diagram, which is called the wedding cake, which shows that the objectives connected to the biosphere at the base. Life on Earth, life at sea, clean water and sanitation and environmental action, they are essential for survival. Without these four green objectives, so to speak, without the environmental objectives, there is no way society can sustain itself. To reach zero poverty, sustainable cities, peace, justice, clean energy, health and well-being, quality education, gender equality, and zero famine, this is very much based on healthy biodiversity. The life support system must be well-based. And if society works, who knows, if the economy will have a greater chance of working. Infrastructure, reduction of inequities, consumption, and sustainable production, and all under a scheme of partnership and cooperation among the countries. The richer countries need to collaborate with poorer countries in order to reach the goals. Because no one is sustainable on their own. If our neighbor is sustainable, we are, and we are not if they're not. So what happens is this. When we look at these agreements, speaking here as biodiversity as the basis for everything, biodiversity renders a service to us. It sounds too utilitarian, but for want of a better term, I'll show you the ecosystemic services. Just in Brazil, what services are these? We have 140 agricultural cultures, a renowned name for its agriculture, biofuels, commodities, biofuels, you name it. Brazil produces. We have 140 agricultural cultures, 85 humming plants in agro systems. It's a great diversity of food sources, fibers, and fuel sources, which is unequaled anywhere else in the world. 245 plant species nowadays used as a base for cosmetics and pharmaceuticals. We have over 500 sacred natural sites. 274 languages are spoken in the Amazon region. Just to give you an idea of how natural and culture are connected. Our expressions within the sacred are very much connected to nature. They come from our tradition with the indigenous peoples, the tradition of the African peoples that lived and live here, and the tradition also brought to us from many European peoples and left their descendants here to this day. So this tradition, this natural service, even a cultural service is rendered to us. And look at this, 80% of water, a great part of our energy is generated from water plants, hydroelectric plants. 80% of the water that supplies hydroelectric plants comes from protected areas in Brazil. So nature, for energy, we depend on nature as well. So, it's not one or two little bits on the side that nature offers us. And when we look again at these goals, nature is at the base of everything, but are our social and economic actions, have they been favorable to the same nature? And that is what our internationalization project at the post-graduation course in ecology here at the UFRJ, their whole internationalization project is geared towards sustainability. And in post-graduation in ecology, we have a sub-project which deals with synergies and trade-offs among these same sustainable development goals. Do they all harmonize with each other? And what we have seen in the surveys in our group is that the answer is no. Not always, right? Synergy. Let's see, when we see our colleagues here making their presentations after me, earthly biodiversity, which is objective number 15, conserve it, has great synergic connection with climate action. the more we want to restore, we are contributing towards mitigating greenhouse gases and adapting society to a climate that will become warmer. So there are synergies, but there are also great trade-offs. Look at objective goal 8, economic growth and dignified work. Economic growth, as far as we have seen in modern age, recently there was a study showing this, showing that it trades off with social equality. Poverty has been reduced greatly in the world in the last 30 years, but social inequality has increased. So what's the use of growing economically and getting worse socially? So the increase in greenhouse gases contradicts the other involvement. Economic growth does away with nature, pollutes the sea. So it kind of gets in the way of objectives 14 and 15. So how can we make the economic growth goal? How to make it synergic with these? Well, I think that is a political, scientific, global issue that is very important. But the fact today is that there is a trade-off. So the path for the SDGs is the path that we want to tread, for sure. And also in the master's thesis recently, in our postgraduate course in ecology, connected to our internationalization project, we see that there is a set of other worldviews that are not necessarily views of sustainable development. So in the global north, people are already talking about not growing or growing backwards. So we're growing, and inequity only grows. Income is terribly distributed throughout the world. So it's about growing and distributing wealth. So these authors preach the degrowth, as it were. There is another movement, ecofeminism, which follows the same line, along the same line, with the importance of women's roles in our reapproaching nature. In the global south, there is a set of groups, Ubuntu, Swaraj in India, Buen Vivir in our Andean neighbors, Ubuntu in Africa, Bolivia, Ecuador, Buen Vivir, Tecoporã, which is a Guarani worldview. This set of visions or views do not have their pillar in economic growth. And more interesting so is that when you constitute a legal matrix in their countries, it doesn't really happen. Nowadays, Ecuador and Bolivia have the law of the mother nature. People can be sued if they impact nature too much. This impacts the ethical view and views of justice in these countries. And this is growing worldwide. We do not have this logic yet in Brazil. The Doce River recently, because they were impacted by mining, they sued the state. They can't do it, but the third sector organization said, well, a river can't talk, but we can talk on behalf of the river, and we are suing the state. The suit was unsuccessful, but others will come that open up this precedence in order for us to change some of these things, and we can have a human as well, not only human ethics, but including other elements of nature. And if that is what will be done, well, I think this horizon here of the sustainable development, it's very important that these convergences have been created in politics. We talk about development and environmental policies. Sustainability brings both together. People say we are social scientists or economic sciences. In law, there is human ethics. There is a discussion about the intergenerational importance. The youths are claiming their future that we are taking away from them. So when they join together, we have the right to sustainability. In our modern world, when these things come together, we get nearer this sustainability. Is that enough? The impression I get is that these discussions among us modern people are excluding other worldviews. There is this the discussion by these groups, Ubuntu, Buen Vivir, Amazonian, and indigenous people. Well, We need to dialogue more with these people so we can bring these world visions to this discussion. Science is a way to interpret the dominating reality, but we have to interact with art as well, with the indigenous knowledge, indigenous practices, practices in urban practices, rural practices, and urban and religion. Why not? Many religions. So to give us time to reach deeper sustainability through an ample dialogue among these many voices, some of them not heard today. And to reach the end of my presentation, I'd like to give you two examples of theses that were concluded in our course within this internationalization realm. 3 years ago, these two studies were launched. Just look how interesting this is. We have these different colors of different biomes in Brazil. Biomes are ecoregions in Brazil, right? We have the Amazon in dark green, in the orange is the Cerrado, Brazilian savanna, in yellow the Caatinga, the semi-desert region, the Pantanal, the marshes, the Pampas in the south, and the green is the seaside. Everything that is in gray in Brazil, we see that there are 20% of the poorest municipalities in Brazil overlapping with the greenest municipalities. These are very poor municipalities with a lot of vegetation. 7% of Brazilian municipalities, that's very little. But this 7% holds 42% of Brazilians' native coverage. 52% of people who live in this area are poor, according to the UN. 13% of the poor in Brazil. So if for these people to leave poverty, you need to deforest, as in the rest of the world and Brazil, this country will lose 42% of its native vegetation, almost half. So our work here is to work together. Scientists, managers, and people whose life needs to improve. They need to work together in order to create income as a mean of subsistence with what exists, not at the cost of nature. That's what when I talk about synergy and trade-offs. How do we turn this into synergy and no longer be a trade-off as it is? On the other hand, there is a red poverty in Brazil as well as the green poverty. Here in green, the richest people in Brazil, São Paulo, Rio de Janeiro state, or here in the southeastern corner, Minas Gerais state, everything you see in very red are municipalities that lost a lot of their vegetation. But these municipalities have the lowest HDIs in Brazil. Poor without internal nature. For these people to leave nature, we need to recover all the lost nature. So once again, we have to find synergy between recovering nature and generating income. Our students have been working with this. We're going to hear some of them speaking. And here I would like to end up with this vision to reinforce how much I think art and other views that are not just scientific are important in this dialogue. This painting inspires me a lot. I'm from Rio, so I'm a bit biased. This is called Rio, I Adore You, I Adore Your Happy People. This painting conveys great happiness to me, and I think it comes from a mixture of green and blue and the mixture of people, technology, planes, cars, different cultures. Through different religions, we have samba, football, we have a lot of cultural manifestations, and all mean life. So in my view, human beings can indeed reintegrate to nature, and I hope that the future of our students and all of you who are watching this event can be as happy as this painting by Lia Nitarachi, a naif painter in Brazil. Andrew, thank you very much, and I pass the floor back to you. Sorry if I've gone on a bit much. So now I give the floor to my colleagues. Thank you very much, Fabio. I'll speak Portuguese now. I really enjoyed your presentation. I'm not in the least bit surprised. It's always a pleasure. I'm always learning with you. And I think you know, we always put forward to the biologists, for example, the cultural issue. There are two views where poverty, internal nature are, reinforced. And what happens when you take away the green and the poverty remains, as has happened in the southeastern area of Brazil? So I think it makes very clear, that there is hope, and the idea that we work best together. I'm always optimistic, almost always, and I feel that we are close to a great change. It could maybe, if anything good has come out of this pandemic, that it be this, that people work better together and we think with more solidarity, in a more solidary way. And, a lot of people are in need, right? I saw in the press the other day here that there were 80 million people needing help in Brazil, 80 million people in extreme poverty, and there were already 90 million people helping out. And that made me think, there are still 80 million people in need, but 90 million helping. So that's it, really. If we can take this to the right environment, we are on the right path. I will leave the questions to the end. There are a few comments as well. And I now give the floor to Professor Adriana Hemley. Professor Hemley, as I said, is at the forefront of knowledge, science. She works with the genetics, the genetic code, the gene code, what happens at the molecular level, and how this can be applied towards the improvement of sustainability. So Professor Adriana, it's a pleasure to have you here, and I now give you the floor. Thank you very much for the invitation. It's a pleasure for me to be here with this panel, all the speakers. I will share with you shortly. Here we are. So that it looks clearer. So I was invited to talk to you about CAP Sprint activity that I was also part of, but within a context which is a very comprehensive project, which is the Medical Biochemistry Institute of UFRJ, which is to generate better adapted plants, better adapted to environmental stress and climate change, and that can keep or increase food productivity worldwide. And why is this theme so important? It is estimated that up until 2050, the world will reach 9 billion inhabitants. We are currently 7.5. So it is a great challenge for agriculture, because it is estimated that till 2050, we will have to increase around 70% the agricultural production at worldwide level. We have 30 years to reach that. And before this problem of intensification and climate changes that bring about great losses of agricultural production, productivity could be reduced up to 70%, especially in droughts, which is one of the main problems in climate changes, agricultural changes.
In parallel, the foundations of our current agriculture wants to increase productivity through practices that bring negative impacts to the environment, such as the intensification of chemical pesticide use, irrigation because of the droughts, and the water is also a problem of the century, and especially deforestation in order to increase the areas that can be agricultural. So the great challenge I consider we have in studying agriculture and production of food is to develop innovative biotechnologies to serve the demand for food in parallel with the preservation of biodiversity. Our plant molecular biology lab at UFRJ has been working for 30 years, and we study the growth of plants and how it is modulated by the environment. And why does that happen? The shape they have, the way they can respond to the environment is in intimate contact with the environmental signs. Just a minute. Okay, so we have various environmental answers, and the way they do it is by modulating the cell growth, the cell divisions. They are regulated through a very sophisticated biochemical mechanism that we've been studying for 30 years. And we've been studying how the environment regulates the cell division. If the environment is favorable, it promotes the cell divisions. However, when the environment is negative, they stop the cell divisions, and the plants hinder the productivity and the creation of foods. And we've been studying this in our lab. And because we have a basic research discovering the biochemical processes, the question is, can we manipulate plant developmental controls to generate change? In this initial project, we work with a special mutant called the AIP mutant. We have many genes that are identified in our lab. And if we manipulate these genes, we can increase vegetable productivity. So AIP is one of them, and we work in the lab with the model plant Arabidopsis thaliana, which has a short life cycle and accelerates our research time. This is a regular plant, and this is the manipulated plant here, the AIP, where you see an increase in the number of leaves and the number of fruit, and the amount of fruit and then the roots. And when we remove water, they tolerate the stress of drought. So in addition to an increase in biomass, they consume less water. Last year, we patented this plant. And in addition to the increase in biomass, these plants are healthier, and this boosts the seed productivity. So the Arabidopsis here produces more than 70% of seeds compared to the average control non-modified plants. So in a partnership with Embrapa, we tested this technology in plants that have an agricultural importance in Brazil, such as soybeans and cotton. And we validated this technology. We see an increase in productivity of soybeans that are around 94%, and in cotton we observed up to 100% of productivity increase. And in addition to that, we use the classic genetic manipulation of plants. What does the Sprint project propose? The main goal of the Sprint project is to develop biotechnological tools that everybody has probably heard about because it received the Nobel Prize, using gene editing CRISPR technology, and intending to increase plant yield. So this Sprint partnership was established with the Center for Plant Systems Biology in Belgium, coordinated by Dr. De Kinsey. We also established, we counted on the PBV, the Program for Biology at UFRJ. We worked on this gene. And what did we do? How did we do it? What's the advantage of this technology? It's genome editing technology with extreme precision. So you can cut, this is the representation of a DNA, precisely, and to modify what is there, the DNA of the plant itself, and insert new elements. And then the result is that the plant acquires a new feature, but it remains as the same species and technically the same genotype. So this is this process is considered natural because it happens in the evolution of organisms. And what we are doing through this technology is to accelerate this natural process, the adaptive process of the plant. And what are the greatest advantages? This can help plants to adapt more quickly to new environments, because this is what is going on currently with the climate change. The climate changes are so quick that they do not give the plants time to adjust and adapt to new environments. And this is really dramatic and really concerning. So this genome edit technology enables us to accelerate this process, to adapt the plants to the climate changes and adjust their own characteristics. That's what we call precision agriculture, using the plant's own genes or wild species. During the mission, I was a researcher, as a senior researcher, and I worked with corn. It was a really pleasant time of my life, because I worked hands-on to characterize the edited plants, the corn plants. I worked in the genotyping and phenotyping processes for maize. This is wild maize. This is the edited maize. It grows more, it has an increase in biomass. And we participated in the work with various lineages. Some of them are more promising, that have a greater increase and more growth in very young maize plants. Our Sprint partnership goes on with the possibility of having students, new students, doctoral students, postdoc students, and we hope they will continue the analysis and use other genes at UFRJ. We have access to a very sophisticated technology to analyze the phenotype of plants. So this is a watering system where we test the tolerance to drought, and it's completely automated. And to conclude, what will be the desirable tomorrows, the positive, what we see as a positive thing, is that this technology is seen as a very positive outcome for world agriculture. We brought this to UFRJ, and we want to train many people with this on this technology, and it has wonderful effects on environment, on the environment and social impacts, because it's really cheap, really cheap. And the time to produce new plant varieties is shorter, and this makes it accessible to smaller to small farmers. It doesn't have to be applied to the large crops, but it can be used for family agriculture, with local farmers. It has a very positive impact on sustainability because what we created with our gene increases the yield and reduces deforestation as a result. And we reduce the use of water, and the plants are more resistant to pathogens, and we reduce the use of fertilizers and pesticides. And to conclude, I think it's a very relevant point to highlight in these studies. I would like to remind you that today, one in every 8 people in the world is malnourished. So if we increase the productivity of food, this is a way to offer hope to these people. So finally, I thank you. This is my team in Belgium. Here, thank you to Carlo Ferreira, another researcher who was in the print mission. This is the director of the institute. This is Mark, our reader and our PhD over there. He is still very active, even though he is retired now. This is the entire team of the research group in the center. Here we're relaxing in an informal moment, and I must thank our staff in the lab. This is unfortunately, I apologize because this is a kind of old picture. It's not updated, but this would not be possible if we didn't have this group of students at UFRJ. And we thank all the agencies that have supported all our research projects. Thank you, Adriana. I think I'll change a little bit. Your talk was fantastic. It's an example of the excellent research work you do. We had to choose among 60 people who had projects in the print program for the Desirable Tomorrows. And I see Fabio nodding here. The impact of your research is enormous. If you use the same plant and you double the yield of cotton and 70% of soybeans, you no longer need to deforest. You can double the production with no need for deforestation. Unfortunately, one of the issues is not just production, but it's transportation. But the potential is absolutely fantastic. So, what you do as a researcher, I have no problem to mention your research with the knowledge you have on bio and technology, information technology, etc. You're doing a wonderful job. I will open for 2 questions by Ana Célia, who's not in the biology area. She wants to know whether after modifying, are they reproducing regularly? The difference of genome editing is not genetic modification. I think that's a very specific concern. It's like a classic monitoring that accelerates some crossings that take 10 to 12 years. So these seeds reproduce. So over the time I was there, I did some cross. I have pictures over there. So yes, the producer can create their own seeds and continue to grow their crops. They're not sterile. The plants are not sterile. The technology does not create sterile plants. It's same with cotton and soybeans. And there is another question here. Oh, well, it's the same question, I see. No, you don't have to buy anything. Once they're growing, I will continue because the time is running. But in the end, you can talk about how you can do, how you can conduct your work together with the social aspect to create the desirable tomorrows. I will start by calling Michelle, who will talk about photosynthesis and solar energy. So thank you, Michelle. The floor is yours. First of all, I'd like to thank you for being here. The invitation was an honor to me. So I will speak about the work I did with the grant from Print. And I would like to give you a more general scenario, not just focused on photosynthesis. I just need to load the full screen, just a minute. My study is on photosynthesis, but it is related to coral reef systems. So I've been working with coral reef systems since 2014, and in the last year of the Sandwich Doctoral Program, I studied photosynthesis and how this affects the coral reef. The coral reef ecosystems are the most diverse marine systems, and they sustain the economy of various places that depend on these systems for fishing, tourism, human resources, and they create billions of dollars every year. This is a very important system because its structure is very important because it is they also they're formed on structural coral reefs, and the environmental damage caused by the loss of these systems could cause huge destruction. The coral supplies carbon gas, and the microalgae produce the synthetic products for metabolism, and this extends to a point that 90% of the nourishment of corals is achieved by photosynthetic processes. In the marine environment, the more you increase the depth, the more you have a light. So because there is a 3D structure, you have an evaluation from some micromoles of photons to 95. This graph, you can see, represents the coefficients of light. And in a coastal environment, you have more variability than offshore.
In the case of photosynthetic organisms, as is the case of the little organism inside the coral, this rate can show drastic change. At an optimal moment, photosynthesis would occur normally. You would have the light energy going in, production of sugars and oxygen necessary for the growth of the algae. But as we are in a reef system and you have a high gradient of light variation, luminous variation, the algae ends up having to deal with an excess of light, because there is no direct relationship between increasing the amount of light and how the photosynthesis can be carried out. There comes a saturation point where the system cannot deal with the energy that reaches this organism. This can induce the production of oxygen reactive species that cause DNA damages and all cell signaling processes. And there is a problem with the photosystem too, directly related to protein D1 of the system. As this protein is degraded by the light system, then there is an inactivation of PSII, thus reducing the efficiency of light use. The maximum of radiation during a one-day period happens for around midday. At this point, photosynthetic activity is lowest because there would be greater stress for the algae. It would be under great stress. So imagine energy reaching the system like a funnel, and then you have damage occurring in this ecosystem. If the recovery rate of the system is not fast enough, it is not able to deal with the energy that's arriving, and there is pressure accumulating on this system. When you have some environmental variation, this can lead to the rupture of the symbiotic relationship between the microalgae and the coral. When this happens, we could have a decrease in the coral tissue growth, calcification, fecundity, and increasing the risk of diseases and mortality. This is very important because global whitening events are getting more frequent. As you can see, with the ocean temperatures increasing, the whitening, the bleaching, sorry, started happening in 1998, and the other one already in 2010, only 12 years later. Recently, we had the third global bleaching event, which happened in 2015. So the interval between these events is being reduced. So the system is at the risk of collapsing. As light is one of the main factors that can affect this symbiotic relation, we have an ideal condition in terms of the symbiont condition, where it is neither concerned about the reparation costs of the system because of the high light intensity, but is not being limited in terms of growth because there's not enough light for photosynthesis to take place. So the optimal condition would be a midway for the functioning of these organisms, and the functioning of the coral were maintained, and they would not starve, as it were. So we carried out an experiment basically focused on the microalgae, and we wanted to understand how the exposure to different radiances changed the growth of the algae, how the photosynthetic efficiency would change during the day under different levels of light, and we described the light phenotypes of symbiogenesis under these conditions. So we chose conditions ranging from low light to high light, and then we have these light angle profiles. In the graph showing the light varying along the day, 12 hours dark, 12 hours light, and the lighter hours, the light would increase and decrease according to sunlight. We had the three main species we worked, Symbiodinium natans, Briviola minutum, and the last one. And in our, we used a control system called Arduino, which is a free system where you can create program codes for your system to function the way you want it to. In this case, we created a code so that exposure to light would be gradual with different peaks. So here in the results about the algae growth, we saw that duplication time was drastically affected in some species due to the low amount of light, would be the condition of green light in the graph on the left, where the algae took around 138 hours to duplicate. And this compared to the intermediary conditions where it would duplicate in 30 hours. Just like with the species, these some variations vary. For the first species, this made no difference, the third amount of light. But in the last case, it was notable, the difference. The second part of my experiments has to do with pressure on the photosystem, where we measured photosynthetic efficiency hour by hour from the moment we turn the light system on, and we can observe that the peak in terms of the minimum efficiency went to the maximum radiancy peak. So these results are two species in this case, right? For both species. This study is very simple in a way, but it's very important for the beginning of this research of the modeling system. And the main highlights here are that we saw differences in the growth of species in light conditions, which can indicate physiological variability that can be associated with the host resilience when associated to the coral. Low light condition could be limiting growth due to not enough light to allow the optimum photosynthetic performance, and higher light could be limiting growth due to damage to photosystem 2. And in this case, the cell would be just investing its energy in staying alive. There would be no liquid photosynthetic production to be passed on to the coral. And in this way, the coral could have its nutrition impacted. This study is a subsidy to the ecological models here in the laboratory I worked with in Pennsylvania. It's being developed there, especially in radiance terms. For the future, we have the objective of understanding this variability, this physiological variability better, to elaborate detailed experiments in order to explore the turnover rates of D1, analyzing its time of damage and repair, and to start approaching light and temperature combined, which are the first factors that have to do with the relationship between the symbiodine and the coral. I'd like to finish off by thanking the people from the lab who welcomed me at UFRJ here, CAPIS for the funding, the genetics program, and I'd like to thank this conference also for allowing me to present you my work. So that's what I had to say, and I'll take questions if you have any. Thank you, Michele. You know, right, that not everyone watching this knows that coral reefs are like the tropical rainforests in terms of biodiversity. They are the breeding ground for so many fishes, and they are suffering a lot. At this point, the whole world is trying to understand all the principles in order to try to improve this. And you see once again how animals in partnerships with bacteria and the photosynthesis, we see that the issue of biodiversity is symbiotic. I'd say it's a cultural issue that Fabio drew our attention to. We're all in this together at the micro to the planetary level. We're all one planet, and within this planet, all cases are connected. I think this work is very important. If we are able to protect and improve, we can reach the point that Adriana has reached, where we help corals and their symbionts who fixate energy, help them to better bear these climate changes. So thank you very much, Michele. We'll take questions at the end. If it's possible here for us to share this, Bruno is having some technical difficulties to come into our event, and we're still within the sea environment. That's what why I wanted to put him up next. The bacteria are within the other symbionts. For those of you who are not in microbiology, most bacteria are good. Sometimes we think of bacteria and disease. No, they do everything. They are the glue of life. They're invisible. They fixate nitrogen, release other elements to other larger beings, be they plants, animals, fungi, all other kinds of types of life. So if possible, we 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 can we put our next speaker on? No, or I think I'll have to give the technician. Can we put Bruno's video on, or we'll have to move to Viviane? I don't think so. Viviane, are you ready? Great. So Viviane will now talk to us. We're going to go out of the sea for a while, and now Viviane is going to talk to us about restoration. We know that there's been a lot of damage, but we also know that how to restore. So you have the floor, Viviane. Thank you, Andrew. Thank you, everyone. Good afternoon. My name is Viviane Dib. I'm going to talk to you about the work I developed, the study I developed during my doctorate at the King's College London, UK. My supervisor is Bernardo Strander and Mark Mulligan here in London. And the title of my work is to include water resources in the prioritization of areas for the restoration of the Atlantic rainforest. Well, this has to do with spatial planning. It's strategic planning that seeks to show cost-effective solutions for forest restoration. Here we are applying this to the Atlantic rainforest in Brazil. And the areas, if they are restored, they would provide more benefits at lower costs, lower restoration costs. This exercise has already been carried out for the Atlantic rainforest, carried out in Nature, Ecology and Evolution publication magazine. When this was carried out for the first time, the criteria, the benefits that were sought for the restoration, which were used to point out these areas, were just the conservation of biodiversity and carbon sequestration, which had to do with the climate. And the challenge after this first stage, which was a landmark, very important landmark, would be the conservation of water resources as a criterion for this prioritization. So with regard to the step-by-step of this methodology, it's a space modulation and algorithm. I won't go into the detail of the methodology, but I just want to show you what was necessary for this prioritization to happen. The first step would be to define the targets. In other words, the area you want to restore in a certain region. In the case of the Amazon rainforest, our area to be restored, our goal was based on data from the biome legal reserves, around 5 million hectares. Later, we define the criteria for this prioritization. Here, we will work with the conservation of water resources and cost reduction for restoration. And then we go through a stage which consists of preparing the database, which will feed this modeling, this algorithm, which would then, with these layers or surfaces, spatially explicit, which show in the case of the Atlantic rainforest biome, the areas with higher and lower costs and the areas which could bring more or less benefit with regard to water resources. And once these surfaces or spatial layers are determined, we can run the modeling and generate prioritization scenarios. And within these scenarios, we can quantify costs and benefits and compare analyses, understand which are best, understand each situation. So the criteria we used in this exercise in my doctor's thesis was on one hand, cost reduction, and there we use the implementation cost for the restoration, which mean the actual costs necessary to plant seedlings, transportation, land acquisition, direct costs, and the opportunity costs, which represent profit that a landowner would not receive from the point that he transformed a piece of land which was destined for pastures and agriculture into an area of restoration. And it's important to put this at the end, at the point of our scale, because the opportunity costs in all this are very high. And these areas, since they provide a lot of profit to the landowners, they would not really be destined normally for um, for this kind of study. So we can see the land use conflicts here, something which already has been talked about in the previous presentations, the need to minimize conflicts, the understanding that we can produce and conserve and restore at the same time. If on one hand we had the costs, on the other hand we had the challenge of putting the conservation of water resources on this scale. So this is what I went to do in London. My international partnership was with Professor Mark Mulligan from King's College London. Professor Mulligan and his team developed a software which is a hydrological or water model called Waterworld. It's free software, so anyone can access it, and it works as a support for decision making and to elaborate public policies, because within this water model we can provide restoration, deforestation, or climate change scenarios, as well as others, and understand the consequences of these scenarios to water variables. And thus, we were able to include the conservation of water resources onto this prioritization scale. So one water variable that the water world model calculates is what we call the anthropic water print. With relation to water quality, this is represented by impact. Different land uses associated to man, such as urban areas, pasture, mining areas, agricultural areas. So the impact of these different land uses and their impact on water quality. So the higher the water print, the lower the quality. So we wanted to understand the water footstep. How would it impact the Atlantic rainforest, which is the focus of our study? So in red on the map, the closest point to the red, the greater the water footprint, so the lower the quality of the water. Then we needed to understand the impact of forest restoration and its impact on water footprint. So it has to do with sensitivity of this and towards restoration. So we simulated the restoration of the biomes, pasture, agricultural areas, etc. And we understood how the response to the water footprint would deal, so to speak, with the situation. The areas in red or closest to red or orange are the areas where forest restoration contributed more towards the reduction of the anthropic water footprint. So this fed our mathematical models, and thus we were able to generate the map of priority areas for restoration, considering also the simple resources, the water resources.
Now on the right, you can see the map, and we have the original area of Mata Atlântica, and we see in purple the priority areas for restoration if we consider water quality alone. if we compare the scenario to the control scenario, which is as if we had restored the same amount of water, the same 5 million hectares, but with no planning and distributing the restoration throughout the Mata Atlantica in a uniform way, this would increase the quality of water twice as much, but it costs approximately 111 billion reals. And we know we have limited resources. On the other hand, we have another scenario that focuses on the cost reduction. And if we see the areas here, they are different from the areas that we showed before. In this model, the algorithm is seeking areas where the cost of implementation is lower, and where restoration would imply lower opportunity cost. So it would reduce the profit that is related to agricultural and cattle activities. However, this reaches only 38% of the maximum potential of water quality improvement. The maximum one was the previous scenario. This one yields benefits that are only 38% of our maximum potential, but it costs less than half. It's 58 billion reals. And the idea is to reach a compromise scenario that would use both weights in the scales, both cost reduction and the conservation of water resources. So this compromise scenario guarantees 55% of the maximum potential of improvement in water quality. So it's a little bit more than a little bit over half of the scenario that considers the improvement of water quality alone. However, it costs 58 billion reals, so it's slightly more than half the amount. So this makes the restoration projects more viable. We must take into account the social aspect as well when we think about the ecosystem return. We cannot disconsider the cost for restoration, and this was our greatest goal to use both criteria. And to conclude very quickly, I want to show some how this tool for spatial analysis can support the decision-making process regarding forest restoration. When we approach the preservation of areas, we want to choose the areas because we can't restore all the areas. We want the choice to be as little subjective as possible. We want it to be based on technical criteria. And this will help the design of plans of water restoration and the payment for environmental services, the compensation areas for environmental licensing processes, and the decrease in the conflicts, the reduction of conflicts because of the land usage. And at the end of the day, this helps us protect our native vegetation and the fulfillment of the SDGs in Brazil. And we can talk a little bit more directly about SDGs number 15 and 6, which are the focus here. We are trying to show the priorities for these two goals, but we know that the restoration strategies and the projects bring about numerous other benefits that are not accounted for here, such as carbon sequestration, well-being and health of the rural population, reduction of poverty, because the restoration chain creates jobs and income for the people who are working on restoration, and many other goals. So I would like to thank you again for the opportunity to develop this study together in partnership with the people at King's College. And I believe in the power of these studies and the ones that have been presented today and the other days. And I do believe in a better future, more based on science. And I think this is my final message. And I'm open to answer your questions after the end of the presentations. Thank you. Thank you very much, Viviane. I think your presentation was excellent. It brings another dimension and scale. And it talks about another moment we are undergoing, the information technology that has advanced and mathematical modeling. I think it's fantastic. And we seek the more efficient way and costly. And when I was looking at you, I thought it costs 58 billion people. It's a lot of money. But then I think, well, 50 million people, if anyone gives 10 reals, it's done. I work with microbiology. It's a very small world. And your research, your study has the scale, and this is why it has great impact. So congratulations. So I will pass the floor to Marina very quickly. Marina, are you there? Yes, she is. So the floor is yours. I'm back. I was off very quickly, but now things are working here on my side. Can you see my screen? Okay. First of all, good afternoon, and thank you for the invitation, Andrew, and all the staff that organized such an interesting event with such a different vision and perspective that we are used to having. I'll talk about my PhD project that I developed in Australia through the Print program. And the title of my project is Estimating the Potential of the Conservation of Biodiversity in Private Areas in Mata Atlântica. My reader is Renato Prozelis. I am at the Ecology PPG, and I'm also connected to another group at the university, at the Catholic University. Our international partners for this project were Dr. Rebecca Runting in the Geography Department at the University of Melbourne and Dr. Hawthorne Bayer from the University of Queensland of the Centre of Information for Decision Making. Both are experts in spatial analyses, especially the ones that are aimed at spatial planning for sustainability, both in terms of land use and facing global changes such as climate change. As Fabio spoke so well about it, we're undergoing what we reckon is the sixth mass extinction of species, and we have been organizing to create targets so that the countries can reverse or at least to stop all this. And probably the goals that are highlighted are the SDGs, and we work with land ecosystems. But as Fabio said, nothing is isolated. We're talking about the water life we're talking about climate action and quality of life and poverty reduction. In Brazil, the scenario is a bit complicated, not only in Brazil, but in many developing countries, because we must develop without degrading, and we lack integrous areas. We have a very high level of degradation, especially in Mata Atlântica. We have very few integrous areas that are not degraded. So let's increase them. However, we don't have large areas to protect in Mata Atlântica. And even the existing areas, we have studies that show us that many of them are not effective in terms of protecting biodiversity, be it due to lack of resources or because they were created on paper, but it's never been fenced, or they had no conservation plans or managers, and sometimes they are created without a study behind it to base the conservation on. So understanding this is crucial, because the goals are not talking about protecting areas, but we want to protect species as well. Protecting empty forests does not solve our problems, or in case we protect the same species all the time. So answering the questions is key to change this scenario. So we come to a question that is also a central question for the ecological theory. Where are the species, and how are they distributed in the space? We have Mata Atlântica in Brazil. It's a critical scenario in terms of degradation. Someone asked about the recent estimates of how much of the original Mata Atlântica we have. That's about 23%, even though we have a more pessimistic estimates that state something about 7 to 10%, it depends. But the most recent one says it's around 23% of original Mata Atlântica. And the place where we have the smallest areas are in the northeast region. Mata Atlântica, in addition to being very rich in terms of biodiversity, but it is also very rich in impacts. It is a coastal area where more than 70% of the Brazilian population lives, where we have the largest urban centers in the country. So to us, it's an open space laboratory, and to understand how we can change all these processes. But of course, it's not feasible in terms of resources, both human and financial resources, to have data on diversity for the entire Mata Atlântica area. So we use technological resources to try and estimate how much biodiversity exists in the areas we have no information on. In the case of Mata Atlântica, what we have left of Mata Atlântica outside the protected areas are small fragments in private areas. So we want to understand how this happens, and this has a lot to do with the changes in the Brazilian legislation defining what has to be protected inside private areas. Oh, and just a reminder, my research group is Synthesis in Ecology and Sustainability of Mata Atlântica, and this group was created to try and meet the demand to understand Mata Atlântica, how Mata Atlântica works, because we have very rich and very poor areas in the same biome, and it's biologically very varied. So we collected data, more than 80 researchers who work on Mata Atlântica brought together their own data, many groups, insects, plants, vertebrated, and non-vertebrated animals, and each grain point is a sample point that was given to us by a researcher. Some areas are not very well covered, unfortunately. We have all these bias that have to do with where the research centers are located, but the ones that have the greatest coverage are the ones we have more samples of. So what do we do with that? We join the biological data, many variables, social, economic, and environmental variables that can interfere with biodiversity. What can influence in the number of species that are present? So, for example, the density of people, the type of soil, the paving, vegetable coverage. So we have more than 80 variables. And for each one, we have different causes because the environmental processes happen in different areas in the system. And we create a complex mathematical models that will try to estimate as precisely as possible what the wealth of the areas we have no data on are. So this map is not part of my project. It's another study by my reader, and he was trying to map in which areas we have the greatest potential for the forests to regenerate naturally. If we remove the impacts, then the forest will grow naturally. This is what the map represents. So the more strong red, the less the possibility, the smaller the possibility. So unfortunately, working with other people's data is very sensitive. We are treating these data very carefully. But my map will be like this, but instead of the probability for the regeneration, we will see an approximate number of species in those areas. We are working on species that we are working only with species that depend on forests. To talk a little bit more about Australia, the partnership and the time I spent there, and what this gave me, what this added to my research, not only with the researchers who are brilliant, they have an incredible ability in terms of mathematical modeling. And I had the possibility of participating in the national meeting in Australia. Unfortunately, I have no pictures of my own presentations because no one could take a picture for me. So nobody could take a picture of me. But it was really interesting because in addition to presenting my project, I was able to learn a little bit more on how the conservation is done over there. So building up a little bit on what Fabio said, in Brazil, we don't have this policy, so to speak, of bringing the indigenous peoples and the social aspects to the biological research. And this is happening very often there. So for the entire Congress, we saw times that were devoted exclusively to listen to the traditional Australian populations and conservation projects that used the traditional knowledge by the Australian population. So that was really enriching, and it brought me some context that I cultivate, that I have until today. So that's what I had. That was my presentation. That's what my message for you today.
Okay, Karina, thank you very much. Great presentation, very important. I like the fact that you left Brazil, studied with other excellent participants in the world. Brazil has many talented people as well, of course. Within the realm that we are, which is the UFRJ, the issues we're trying to resolve here are global. The solutions are global as well. But I like the fact that showing how Australia actually is trying to restore the damage done to their indigenous peoples. They're trying to kind of make things better. There are some issues that they can learn from them and they from us. Thank you very much. So now we have our last presentation by Bruno Francisco Oliveira. I hope our technical team can put up the video. Sorry, actually, he wasn't able to put up the video, but we were ready for this and we prepared a video. So then we will conclude with a discussion on everything that was seen. So can we put Bruno's presentation up? Great, okay. Yeah, there's no audio. Can anyone hear? We from the interpretation channel definitely cannot. Therefore, there's nothing to translate for the time being. While we wait for this, let's take advantage of the time we have. And we had a question about how much is left of the Atlantic forest, rainforest. I think Marina or Viviane can talk to us about this. What is the current situation of the Brazilian Atlantic forest or Atlantic rainforest, the Mata Atlântica? Marina, Viviane, I think Viviane will be the best one to talk to us about this. Marina talked about this a bit during her presentation. We have different estimates which vary according to the method used to measure the remaining forest areas. Current estimates say 7% remaining, some say 11. Most recent estimates under Professor Fabio's supervision of Camila talk about around 20 something percent remaining. So there's this difference in the southeastern region. There is the Serra do Mar. The Atlantic rainforest is more degraded in the northeastern region of Brazil. So there are differences according to the best preserved regions and the least preserved regions. Yes. I don't know if this is still ongoing, but we did have a monitoring project led by the National Space Research Institute and the Mata Atlântica Research Institute. They carried out surveys yearly saying how much of the forest was left per state, sometimes even per town. The state of Minas Gerais, for example, started losing their Atlantic forest after a while, that they didn't lose anything. São Paulo state is going through a regeneration process more than loss. So there is variation per region. The Atlantic forest is unique due to its characteristics, that everything is latitudinal, right? From the poorest people in the borders with the Cerrado or the semi-arid areas. In each area of the country, we have different standards. Andrew, if I could summarize here a bit what the girls just said, a study was carried out by our post-grad team by Camila Rezende, where she showed through the state-of-the-art technique in terms of resolution, image resolution, that there are 28% of the Atlantic forest remaining. But what matters here is that around 83% of the forest fragments of the Atlantic rainforest have less than 50 hectares. It's like a Swiss cheese, really, the forest. There are small islands of vegetation that are large. That's why this work by the girls with restoration and conservation are very important in order to create links between these fragments that remained. We have some evidence showing that for this, if for this biome, if we can reach 35% of vegetation covering, we will be able to save many species from extinction. So we are very optimistic that if laws in Brazil are adhered to, especially the protection to the Atlantic forest, which is a law that was enacted in 2006, for the protection of the biome, and the new forestry code, which assumes that degraded areas by the rivers and need to be protected. If these laws are enacted or are adhered to, we'll have around 35% of preservation. This will be essential to retrieve or recover all this that was lost. Everyone here is obviously going through the pandemic. Our lives changed over the last year, have changed since last year to now on many different levels. And there's the economic issue that world governments, most of them, have fewer resources. And we have that there are costs, there are restoration costs, even working at in the most efficient way. So this leads to the question, if governments do not conserve because they have other priorities right now, what are the paths to be taken? NGOs? Do you think the NGO's role has been growing? We are going through a difficult moment. What are the possible solutions? You that study the forests, you could tell us, what do you suggest, Fabio, and then Marina maybe, and Adriana? I think it's important to bear in mind that to conserve is always cheaper than to restore. So when we restore after degradation, it's bad business, very expensive. So faced with the degradation of these systems, such as the Atlantic forest and Cerrado, and also the Pampas and the Caatinga, the semi-arid region, what we have remaining is what's left. We need to return the life support that they have, return to these systems. Especially the private sector, I think, has a great role to fulfill. They cannot get away from this debate. The world private sector is responsible for 60% of the world's GDP. And we know that GDP has been generated at nature's cost. So it's time that private sector becomes involved. The forestry code has a lot to do with private property. So the law must be adhered to within private property. And if it isn't, the authorities must take the necessary measures to punish those who disobey the law. This was revised in 65, 2002, and 10 years later, after it was enacted way back in the 1940s, something has to be done in order to retrieve some of this damage that we created, and it's turning against us. We're not talking about, protecting A, B, or C species. We're talking about our own species. Nowadays, cities like Brasilia, Belo Horizonte, Rio, and São Paulo are going through water shortage, as Vivi mentioned. When I was a kid, we used to throw water around. There was no water shortage. Nowadays, Rio de Janeiro, a warehouse for the Atlantic forest, has water shortages. It's going to cost us, right? It's not going to come at zero cost. But money, as we have seen, is badly distributed. It's not that we lack it. one of the things that I like to remember about Rio, I like to remind you, our Tijuca forest has been deforested, and we went without water. Then, you know, it was improved, and then the population grew, and we have economic difficulties. I don't know if we're going to have Bruno's presentation, because it's a great loss. He was going to talk about the characterization of bacteria allied to sponges in the sea. Sponge are interesting animals because they are 60% something else. In terms of species, they are not sponges because they have everything, millions of bacteria included. But I'm going to at least convey the idea. Let's see if it'll work now. Let's cross our fingers. Cross our fingers, open our ears. No. No, I can't hear. No audio is coming out, unfortunately. Oh, is that something? Andrew, are you using the full screen? No, I'm not the one showing it. We had this problem the other day, and it seems that the knack was not to use full screen. And then the sound will come up. Yeah, that's a good tip. Let's see if the technical support team. Yeah, sometimes you have to enable it for the system to capture the sound. Yeah, you have to control the sound to see. Yeah, on the volume. Yeah, usually on YouTube it doesn't work. It's kind of decorative. Sponge microbiology, the title. No audio.
Ouvindo alguma coisa.
Yeah, I can hear something.
Sob a outra fronte, nós temos a exploração biotecnológica desse microbioma associado às esponjas marinhas.
Right, the English booth is just stating that it's not, it's almost impossible. No, it sounds really, really problematic. Professor Laporte has been working for over 16 years with these fantastic organisms, especially the microbiome of sea sponges. Since 2015, now it's got even worse.
Particularmente no ecossistema de água rasa, muito suscetível aos efeitos de maré, no litoral da cidade de Cabo Frio, chamado de
In the city of Cabo Frio, they were very much subject to tide movement.
espécies de plaquinha celulosa, dessa esponja rosinha que vocês podem ver, esse futiculoso, que foi o projeto da minha tese de doutorado. Nós nos dedicamos, nós nos debruçamos a explorar sobre a ótica independente de cultivo. Você não precisa cultivar o microorganismo. A transmissão do microbioma.
No, sorry, we are trying. Believe us, those who are listening to the English translation. I think this presentation would need a translation to Portuguese as well.
Tanto as frentes ecológicas quanto biotecnológicas.
And
esses potenciais biocatalisadores industriais, com propriedades industriais, no procabiótico bacteriano cultivado dessa esponja marinha. Então, nossa parceria internacional me levou a estabelecer um
this international partnership led to cooperation from the Cork University. No, sorry. From the beginning of the millennium, Professor Dobson's team has been studying the microbial sponge biotechnology. How
enzimas capazes de degradar plástico, poliésteres, os mais diversos como prateados. E nesse contexto, eu procurei o professor Lawson, nós procuramos e estabelecemos essa parceria, e a proposta geral do doutorado sanduíche foi particularmente estudar algumas potenciais estirpes bacterianas isoladas dessa esponja bacteriana rosa, só que uma chamou nossa atenção, que foi a estirpe de pseudocromonas, que devorava a água desde quando ela foi isolada. Isso nos chamou muito. Principalmente teve deficiências, poucos estudos de agarases e marinhas no Brasil. E dessa forma, nós estabelecemos, então, um foco. Já poucos estudos também que estavam dedicados ao estudo de enzimas que degradavam esses polissacarídeos marinhos como agar. Nós nos dedicamos a explorar os genomas dessas bactérias, particularmente essa Projeto de Internacionalização, para encontrar...
study the genome of these bacteria specifically. This year, the first part of our work was to try and explore the influence of tides. This sea sponge, part of its microbiome was
procariótica nessa esponja. Então, dessa forma, a transmissão horizontal é a mais importante, o componente mais importante na existência desse microbioma, dessa esponja tão peculiar encontrada ali no litoral de Cabo Frio. Isso nos leva, inclusive, a hipotetizar que essa esponja é muito resiliente e é mais adaptável às condições ambientais adversas, justamente por depender tanto desse fator horizontal para moldar a sua microbiota. E com relação especificamente ao nosso projeto...
With regard to our project specifically, in conjunction with the Irish researchers, we were able to isolate the genome, and many coding genes were discovered. Our structural proteic analysis in silico, no.
E alginato-liases, todas as enzimas capazes de degradar polissacarídeos marinhos como agar, arginano e alginato, nós nos dedicamos a tentar entender, desvendar essas estruturas proteicas com base nas análises genômicas e proteicas em sílico, e descobrimos que 3 biocatalisadores desses, duas agarases e duas alginato-liases, particularmente, possuem uma termostabilidade provável, interessante, e também processilidade, que são muito valiosas sobre a perspectiva de sua aplicação industrial, tanto na biorefinaria, biocultural de terceira geração, quanto na geração de produtos oligosacarídeos com atividades ativas para a indústria farmacêutica, nutracêutica e cosmética. Então, essa esponja também mostrou ter sobre microbiota como uma fonte interessante de biocatalisadores para diversos setores industriais. Então, as nossas perspectivas futuras, que são formando a base de dois projetos importantes do nosso grupo de pesquisa, é a clonagem e expressão heteróloga de caracterização bioquímica de exobeta-agarase e alginato-liases desses tipos de pseudotormonas isolados da proteína celulose. Nós paramos especificamente nessa etapa da clonagem, bem no final do meu doutorado de sanduíche, com o início da pandemia, e atualmente nós estamos tentando, prosseguindo com esses projetos, e basicamente ele vai se identificar em duas frontes. Nós vamos explorar as exobeta-agarases e aplicar as mesmas para a degradação de biomassa de macroalgas vermelhas, para a geração de açúcares fermentáveis e assim viés de aplicação da biorefinaria, e a outra fonte na geração desses neoagrooligosacarídeos derivados dessas agarases, dessa biomassa bruta de rodopícias, para investigar as possíveis atividades biológicas de interesse desses naus, desses oligosacarídeos. E na outra fronte, nós vamos tentar estudar essas alginato-liases como potenciais enzimas para o tratamento de patógenos bacterianos formadores de biofilmes multiresistentes aos antimicrobianos, e dessa forma tentar fornecer uma terapia alternativa ou de suporte para o tratamento dessas infecções bacterianas tão problemáticas no âmbito hospitalar. Queremos agradecer essas instituições que foram importantes para a informação, as agências de fomento, a organização do evento Design Multimores e toda a equipe do UFRJ. Muito obrigado.
Okay, obrigado, Bruno.
Thank you, Bruno. Unfortunately, it was difficult for me to understand what you said, but I could understand the impact of the genetics and the biochemistry that is associated to the technology that protects this animal from the environment and brings some protection. I will ask the speakers who are still at home to turn on their cameras. The questions and answers have been answered, and I let the time go a little bit beyond. But if you have a question, you can ask the question now. Otherwise, I will close the panel. Our panel, climate change, biodiversity, and environmental governance. I would like to thank you all. The time unfortunately, Bruno wasn't here. I see Anna Celia. I would like to really thank her, she organized this event and gave us the space for us to share. I organized it with all the coordinators, session coordinators. So each coordinator must be congratulated as well for the impeccable organization of the sessions, and thank the translators and technical support, because you're part of the team. The communication is fundamental. Without Christiana and Thomas and the other people who are hiding behind the words technical support, you're very important. So it's the team. Hobson doesn't want to show his face. He's doing. Hobson has saved my life at least 10 times, I believe. That's true, Julia. I don't know who's in technical support today, but it's interesting to see you. It was excellent. I really enjoyed it. What I was saying is that in general, you have a more theoretical, conceptual session, and then after that, you have a session of very interesting applications of the discussion on biodiversity, sustainability, climate, and so many themes that are so dear to our universe of sustainability. It was really good. I learned a lot. You know, I do not belong to this area, but I always learn a lot, and I have much interest, because I think there is no theme that is more relevant to this one, which crosses over all the other themes. So it's been very enriching to participate in all these panels. So thank you very much once again. And I hope you participate in the next panel. So I will close. Thank you very much and see you next time. Thank you.
Boa tarde.
Tchau,
Tchau.
Obrigado por sua compreensão. Obrigado. Thank you, sir. All right. Obrigado.